Okay, so let's talk about capillary fluid exchange. Once we get to the capillary level, if you look at the blood vessel, it only has the tunica intima layer. So it only has excuse me, the endothelial lining. So the squamosal endothelial lining. The reason for this is because you need to get fluid to circulate into the interstitium and back again. Why do we need fluid to circulate into the interstitium? Because we have to get nutrients in to the cells and we have to get wastes out from the cells into the bloodstream. So fluid movement occurs via osmosis through the endothelial cells. Fluid movement into the interstitium, and you need to be careful about language, right? So think about that fluid is moving from the bloodstream into the interstitium, and that's driven by hydrostatic pressure. When you think of hydrostatic pressure, I want you to think of the pressure generated from the heart or from the left ventricle, and that would be your mean arterial pressure. Fluid movement back into the blood or reabsorption is driven by osmotic pressure. When you see osmotic pressure, are you going to think water or particles? I want you to think of particles, right? So osmotic pressure is pressure due to particles in the solution. In this case, it would be your plasma or in your interstitium. And that's the oncotic pressure because we're dealing with albumins, right? Albumin is the main particle that deals with or that gives rise to your osmotic pressure. Okay, so filtration is movement of fluid from the blood into the interstitium. This is driven by, again, hydrostatic pressure. It's pressure in the blood vessel or mean arterial blood pressure. Filtration is going to force water and small solutes through gaps between the endothelial cells, and it leaves larger solutes like albumins stuck in the bloodstream. Why do albumins get stuck in the bloodstream? It has to do with the size of them, right? So they're too big, they can't fit through the endothelial cells, or they can't fit through those gaps. So here's a picture of capillary hydrostatic pressure. Orientate yourself. Let me change the color of the laser pointer. We'll do green so you can actually see it. So take a look at the first picture. So inside the capillary, you notice that if you look at the right side, you have amino acids, you have blood proteins, and these guys are big. So what would you guess this blood protein is? Right, so it should be albumins. And then you have glucose, and then you have ions. So give me an ion, like sodium, calcium, what have you, magnesium, potassium. All right, and so notice that these are endothelial cells because this is one cell layer of squamosal cells. It is a simple squamous endothelium, right? This is actually the capillary. So notice how there's little gaps in the capillary or between the cells or between the endothelial cells. Because there's gaps, that is going to allow molecules to flow through. So if you look at the small solutes, they're going to be able to flow through those spaces or those gaps at the bottom picture. Now notice it's small solutes. So what does that mean? What can fit through? So can ions go through the endothelial lining? Yes, they can. What about glucose? Glucose will go through. What about amino acids? Amino acids will be able to slide through. What about your albumins? Albumins are not going to be able to slide through because they're too big. So albumins stay stuck in the capillary. Your red blood cells, your white blood cells, your platelets stay stuck in the capillary bed. Everything else is going to filter through into the interstitium, right? And once it gets to the interstitium, then make a note. Then your cells can take whatever they need, right? And then the cells are going to kick out waste into the interstitium. What forces molecules through the endothelial lining, it's the hydrostatic pressure. So if you look at the top picture, it says capillary hydrostatic pressure, CHP. So capillary hydrostatic pressure is the pressure generated by the left ventricle. It's also driven by MAP, and the capillary hydrostatic pressure is going to force fluid from the capillary into the interstitium. Why does that happen? Because the hydrostatic pressure in the capillary is greater than the hydrostatic pressure in the interstitium, so it forces fluid out. So which way, just to recap, does hydrostatic pressure push fluid? Is it going to push it into the capillary or out of the capillary? Good, it's going to force it out of the capillary. Okay. Reabsorption, capillary reabsorption, is movement of fluid back into the capillary. So the first time we absorbed it was from the digestive tract, right? From digestive tract into blood. Then we filtered it out at the capillary, and now we're reabsorbing it. So it's the second time we're absorbing it. So be careful with the language. And this is movement of fluid back into the capillary. How do we get this? It's driven by a different pressure. It's not driven by hydrostatic pressure. It's driven by blood colloid osmotic pressure, right? Break this down. What does it mean? Colloid osmotic pressure. Colloid is the size of the molecule, and it's an osmotic pressure, so it's pressure due to particles. So it's pressure due to particles that are colloid size. Which particle is that? It's going to be albumins, right? And then what is the particle concentration or percentage of the particles in the plasma, or what is the osmotic pressure in the blood? So it's around 0.9% or 310 milliosmoles, right? So there's a slightly higher colloid osmotic pressure in the blood versus the interstitium. And so if you look at this, whichever side has the higher osmotic pressure, that's going to pull water towards it, right? So which side has a higher osmotic pressure? The capillary or the interstitium? The capillary is going to have a higher osmotic pressure. And why does the capillary have a higher osmotic pressure? Because albumins are stuck in the capillary space. Okay, they can't move into the interstitium. And because albumins are stuck in the capillary space, they are going to pull water towards them because that makes a higher osmotic pressure. Okay, here's a picture. So I like this picture because it shows you the opposing forces. So if you look at the key, the red arrow is the capillary hydrostatic pressure. The yellow arrow is the blood colloid osmotic pressure, and then the gray arrow is a net filtration pressure. So hydrostatic pressure is dictated by what? It's dictated by the heart, right? So you want to think of mean arterial blood pressure, and that's generated by the left ventricle contracting, right? So that's the pressure in the blood vessels. Then the other force that we're dealing with is the blood colloid osmotic pressure. That is the pressure due to particles in the blood. And the main particle in the blood, as we know from covering blood, is going to be albumins, right? So take a look. Here's a capillary bed, or here's a really simplified capillary bed. Here's your arterial, right? And here's your capillary bed, and then your venule side. So here's your arterial side of your capillary bed, and here's your venule side of your capillary bed. So left arterial side, right venule side. Let's take a look at the two forces. So as blood flows through this tube, there is a hydrostatic pressure associated with it, right? So hydrostatic pressure is forcing fluid out of the capillary. So how does that happen? Hydrostatic pressure is higher in the capillary and it's lower in the interstitium. And so hydrostatic pressure is going to force fluid out. All right. And notice what happens as we go through the length of the tube. So this red line, or the red arrow, indicates hydrostatic pressure. So it's 35 millimeters of mercury. In the middle, it's 25. And then at the end, it is 18. So what happens to hydrostatic pressure as you go through the length of the tube? Good, it decreases, right? So it goes from 35 to 18. Why does this happen? A couple of things. So there's less resistance as you go through the tube. You are losing fluid into the interstitium, and so there's less volume, and so there's less pressure, right? And that's the main reason. Take a look at the other force. So the other force that we see is the yellow arrow. What does the yellow arrow represent? It's going to represent blood colloid osmotic pressure, right? So osmotic pressure is forcing fluid back into the capillary. How does that happen? The osmotic pressure has to be greater in the capillary than in the interstitium to get fluid to move into the capillary, right? So what is this telling me? This is telling me that inside the capillary, we have more particles compared to outside the capillary. Why do we have more particles inside the capillary? What particle do we have inside the capillary that can't escape out into the interstitium? Okay, it would be albumin, right? So the major plasma protein is albumin. That stays in the capillary, and because that stays in the capillary is going to have a higher colloid osmotic pressure than the interstitium. So more particles in the capillary, fluid's going to move into the capillary, right? You're going to pull water towards it because water wants to dissolve the side with more particles. So blood colloid osmotic pressure is pulling fluid in. And notice it's the same number. It's 25 millimeters of mercury throughout the length of the tube, right? Okay, so how does this work? You have to take into consideration the capillary hydrostatic pressure and the blood colloid osmotic pressure. Whichever number is greater, that force wins and fluid's going to move in that direction. So take a look at the arterial side, the left side of your screen. So we have capillary hydrostatic pressure forcing fluid out, right? That's called filtration. But then we have osmotic pressure, blood colloid osmotic pressure pulling fluid back in. Which one is greater? The capillary hydrostatic pressure is. So what do we get? Do we get filtration or do we get reabsorption? What do we call forcing fluid out? Okay, good, we call that filtration. So the difference in these pressures is positive 10, right? So 35 minus 25 is 10. And that is positive, so that means it's favoring filtration. Or the net filtration pressure is a positive 10, favoring filtration. But I'm not going to have you guys calculate these. I could, but I'm not going to have you calculate these. But I want you to understand the theory behind this, okay? I want you to draw this out several times. I want you to understand why we get fluid moving out and how do we get fluid coming back in, or how do we get recall of fluid coming back in. In the middle, there's no fluid movement because the forces are equal and opposite. So capillary hydrostatic pressure is forcing fluid out, and blood colloid osmotic pressure is pulling fluid in, and they are equal forces, and so there's no fluid movement. At the venule side, we have reabsorption. How do we get reabsorption of fluid moving back into the capillary bed? Which number has to be higher? Hydrostatic pressure is always forcing fluid out. Osmotic pressure is always pulling fluid in. So obviously osmotic pressure has to be greater than hydrostatic pressure. So take a look. So hydrostatic pressure is 18, forcing fluid out. Osmotic pressure is 25, pulling fluid in. So which one is the greater pressure? Osmotic pressure, good. And so because this is the greatest and it's pulling fluid in, fluid is going to get, well, pulled in, and we call that reabsorption. If you take 18 minus 25, that gives us a -7. So the net filtration pressure is -7. This means that we're not filtering, that we're pulling fluid back into the capillary. So this sort of, this creates a little micro circulatory loop, right? So fluid leaves the capillary bed at the arterial end, and that's called filtration. And then the cells pick up whatever nutrients they need and kick out wastes, and then that stuff gets reabsorbed and back into the venule and then into general circulation. Capillary exchange at arterial end, fluid moves into interstitial fluid because of hydrostatic pressure is greater than blood colloid osmotic pressure. At venous end of capillary, fluid moves out of interstitial fluid because hydrostatic pressure is less than colloid osmotic pressure. Capillaries filter out more fluid than they reabsorb. So capillaries kick out about 24 liters a day, right? And they reabsorb 20 liters per day. So what are we missing? Good, we're missing 4 liters per day, right? The excess fluid, where does it go? It has to go somewhere. So it's going to enter the lymphatic vessels. Lymphatic vessels are open-ended at the end, and they are going to let particles that can't get through the capillary from the interstitium. They're going to let these particles go into the lymphatic vessels. They're going to go through the lymph nodes, and then they get dumped back into ultimately your venules, so your inferior vena cava. This purpose of this is to ensure constant plasma and interstitial fluid communication. It accelerates distribution of nutrients, hormones, and gases through the tissues, and it transports insoluble lipids and tissue proteins that are too big to get through that capillary wall. It flushes bacterial toxins and chemicals to your immune system. Okay, so capillary dynamics. When does capillary hydrostatic pressure increase, decrease, and how does that affect filtration and reabsorption? When does blood colloid osmotic pressure increase and decrease, and how does that affect filtration and reabsorption? So what I would like you to do is I want you to draw your own picture. So I want you to draw your capillary bed. I want you to explain hydrostatic pressure forcing fluid out, and we call that filtration, and then blood colloid osmotic pressure pulling fluid back in, and that's called reabsorption. And I want you to think, what if capillary hydrostatic pressure increased? Would that make filtration greater, or would that make reabsorption greater? And what if capillary hydrostatic pressure decreased? What would that do? Would that affect filtration? Would that increase filtration or decrease filtration? Would it increase reabsorption or decrease reabsorption? This is something that you need to spend some time on and work through it. You need to draw your pictures and understand how you get filtration and reabsorption first. Then you need to think about, okay, what happens if hydrostatic pressure is higher, right? So what if I go running or I'm out exercising and hydrostatic pressure goes up, my blood pressure goes up? What happens to filtration at the capillaries? What happens to reabsorption? What if my osmotic pressure is higher? Let's say I've lost a lot of water. I've been sweating excessively. I've lost a lot of water. And your osmotic pressure is higher because you're dehydrated. How is that going to affect filtration and reabsorption? So take some time to go through that. And this would be something that would be good for you to go over with me in office hours, or virtual office hours, or we will be having tutoring as well. So it's something that you should go through with me for tutoring. I gave you examples. Okay, so dehydration. When you dehydrate, you lose water, and so you decrease blood volume, right? And because you decrease blood volume, you have less blood, so more particles and a lower pressure, right? And so I walk you through these. I want you to go through these with your diagram on your own and see if you can explain it. You can read those. Edema, so when you force fluid into the interstitium, we call that edema, right? And we'll stop there. I want you to work through that before you start this next portion, and then come and talk to me about it if you are stuck.